Hello everyone. For this lesson I want to just do a really brief introduction to CRISPR, which is a new genetic modification technology, and I want to kind of go through the ideas on how CRISPR might function, and then we'll link to some other videos that you can watch that have a better, more detailed look at this. So CRISPR is this newer gene editing technology. It uses what's called the Cas9 protein, which is a special type of protein that can be loaded with RNA. And this is really the key, and this is where I really like I like to discuss this because it fits very well with all of the stuff we've covered already. Protein is something that your body or your cells will make from an RNA code. So if you were to feed a piece of DNA, if you were to feed a cell some mRNA, it might even be able to create this Cas9 protein if you had an mRNA that had the right codons for this Cas9 protein. So you could actually feed a cell some mRNA, maybe injected using a piece of a virus, which can do that sort of thing. Inject it into the cell, and the cell could make a protein. Okay, so now you have a protein. Proteins are good at manipulating and changing chemistry in the cell. Well, the Cas9 protein is very similar to something like topoisomerase or even like a reverse ligase. It is a protein that helps the DNA strand be sliced and diced. It can make breaks in the DNA strand. But to do this, to make these breaks, it has to first match a sequence of RNA that is stored within the protein. This is not too dissimilar to the structure of, say, the ribosome, where the ribosome is made of protein and rRNA. Here we have a Cas9 protein that's going to be made of protein and a strip of RNA that's inside of it. So maybe we should look at the picture of it here. So here is the protein. This blue thing is the protein. So this blue thing right around it, that's the protein. And then inside the protein, there is a strip of RNA that is inside of it. And the trick is that now scientists have figured out ways to change what this guide RNA is. They can keep the sequence for the protein constant because the protein does its job. It makes little cuts in the DNA strand, but they can modify. The modification is that they can change what this sequence is. So think about the implications here. If you can put whatever RNA you want inside of this protein, you can control where this Cas9 protein goes along the DNA strand, because it will only go to the matching sequence on your DNA. So as long as you carefully create your mRNA so that it matches somewhere on the DNA strand, the Cas9 protein will scan across the DNA strand, not too different from, say, like an RNA polymerase that opens up the strand and then keeps it open for a little period of time as it scans across it. But in this case, instead of the RNA polymerase, which allows for the making of an mRNA, this protein scans across the DNA strand and is looking for a place where its own RNA will match. When it finds that match, it will use its sort of enzymic process to cause damage to the DNA strand. It'll cause a breakage. Now, typically, if a breakage occurs in the DNA strand, something like DNA ligase or a similar enzyme will just come along and repair that strand. And it might do that very quickly. But if at the same time as all of this is happening, you were to also insert some donor DNA into the cell and into the nucleus, inject it somehow, whatever method you think might work, something similar to even that DNA bombardment would even work, but in this case they're probably going to use some more specific or more targeted donation, maybe a virus. Again, viruses are good at donating DNA. If a little bit of DNA was floating around at the same time this protein had made this break, then it might be possible that on the occasional chance that DNA ligase would get confused and it would make the repair not to the original strand, or whatever similar enzyme, it would not make the repair to the original strand, but it would make the repair to the donor strand. And the donor strand would be spliced in to that spot. Now the cool thing is that because the Cas9 system uses this RNA, notice now that the RNA no longer will match, because where it made that cut, you've placed some new DNA. So now that you've placed some new DNA, the Cas9 is still floating around. It's still trying to cut the chromosome open, but it can't because it no longer has a matching sequence. It now has a sequence that doesn't match because the donor DNA was inserted. And if you extrapolate this idea a little bit or play around with it, let it roll around in your head for a minute, you'll realize that even if your DNA gets properly repaired, as long as the Cas9 is floating around in the nucleus, it will be continuously cut and broken until the donor DNA slips in there. So until the cell makes a mistake and inserts that donor DNA into the spot of the strand that you made the break, until that happens, the break will keep forming because the Cas9 is floating around. So that's what the CRISPR system is. CRISPR uses a protein that has that sort of enzymic property. It can cut a DNA strand, but this protein has a piece of matching RNA inside. And by controlling the presence of the protein and the type of RNA inside that protein, you can control where the cut happens. And this cut will now only happen in a specific spot on the genome. It will only happen at, hopefully, across all of the chromosomes, because you're hopefully going to be using a piece of guide RNA that is very specific to only a single spot in the whole genome. And so by guiding it to that single spot in the whole genome, you can insert any donor DNA you want, and that will create a new organism or a new gene in that genome. Maybe you're just going to put in a donor that has a broken sequence, just something to break it so that you can stop the gene from being active. Or maybe you'll insert an entirely new sequence of DNA, and that new sequence of DNA will cause the organism to have a different phenotype, something new that it will give it new properties or new abilities. So let's just go through here how it works. So Cas9 is a protein with a designed guide RNAs, which are injected into developing embryos. We have to target the creature before too many cell divisions have happened, so that we'll get a nice clean, so that every cell that divides will have this new phenotype. Once inside the cell body, the Cas9 searches through the cell's genome for a DNA sequence that corresponds to the sequence in the designed guide RNA. So the design aspect here is that you are designing this piece of guide RNA so that it finds a specific spot in the genome. Once the sequence is found, the Cas9 is able to unwind the DNA helix, and after it unwinds it, it makes a double-strand break, a break across the entire chromosome, or the entire DNA strand. Repair enzymes will repair the DNA strand break while integrating any new experimental donor DNA that is present. They might be repaired multiple times, but only on that last time, when you incorporate the donor DNA, will it stop being broken, because the Cas9 will no longer be able to find the sequence that it's looking for. So the combination of having control over where the cuts are made and the sequence that you're donating allows you to have a tool with a very high success rate. And there's lots of interesting achievements that have happened. So I'll go through a couple of examples that have happened. Some of them are already in place, and we'll definitely watch a video, or definitely I'll support you watching a video on the mosquito one, because that one is very interesting and it's already active. That experiment is already going on right now. So CRISPR has helped make the way for several amazing achievements in the last few years. So for example, mosquitoes and malaria, this was a very interesting field. I'm not sure where it's going. There's been a couple of other developments in other gene experiments, but researchers used CRISPR to produce mosquitoes that were practically immune to the parasite called Plasmodium falciparum, which is also known to be the root of malaria. Malaria kills about 500,000 African people every year. So it's a very deadly disease. So they created a method for the mosquito to naturally defend itself from this parasite. To defend itself from the parasite advancing from its blood in its stomach to its salivary glands. So now that the mosquito can't get into the salivary glands of now that the parasite can't get in the salivary glands of the mosquito, it cannot be infected into a human. They additionally made sure that the gene change was inheritable, and that they used a system called gene drive, where it actually, if you were a heterozygote, the gene would actually transpose itself onto the other homologous chromosome, and so you would end up going from, even though the cell should have inherited a heterozygote genotype, it'll actually become homozygous through another technique. There was some other experiments that are being done in pig embryos, and so they were changing pig embryos with CRISPR. They were able to edit 60 genes simultaneously. This is 10 times more than any other gene editing technique that we've ever attempted. And they were able to insert new DNA that causes the pig cells to naturally produce an enzyme that combated the porcine androgynous retrovirus, which is a virus that pig cells may hold that can infect humans. This might be a possible solution to our organ replacement problem, organ transplant problem, in that we could take pig cells that no longer could carry this virus, that would be very deadly to humans, and those organs could then be used for human transplant, hopefully, at some point. There's also a new experiment in HIV and CRISPR. So researchers were able to eliminate the HIV-1 DNA from human cell lines by changing the cells to make them immune to future HIV reinfection. Now, in 2016, the process was not permanent, but in 2019, there was a pair of twins that were born to a couple where I believe the father was HIV positive. So there's a worry that the children would possibly inherit, would possibly become infected with HIV, and so they did this. They used CRISPR to change the children's cells so that they would inherit this naturally occurring mutation that some individuals are born with naturally. So this HIV resistance is actually a naturally occurring mutation that is found in a very small percentage of the population. But in this case, the scientists felt that it was a valuable enough test subject to attempt. So they did this change. It is the first time that genetically modified human beings have been born, and it is very controversial and very interesting. There was the other example where we are very close to maybe moving to a human model, but probably not because of the issues around modifying humans. But Duchenne muscular dystrophy was permanently fixed in mice using CRISPR. This was one of the very first examples of CRISPR being used for this purpose. As much as CRISPR has been studied for a long time as its value to viruses, and this was one of the first examples that it was effective in, and so they permanently fixed the Duchenne muscular dystrophy by essentially infecting the lung cells of these mice with this CRISPR, with this virus carrying CRISPR, and the CRISPR changed the DNA of these mice, and their Duchenne muscular dystrophy symptoms disappeared after, I think it was 75%, over 50% of their cells had to be modified. Duchenne muscular dystrophy is a very tempting one to target because it has a very small mutation that causes the disease. CRISPR has also been used in plant examples, although I believe all these plants are still in the lab stage and not implemented in our food supplies. As I mentioned, there are very few actually genetically modified organisms that you can buy, at least in the plants, even in plants. But there is some consideration for corn and soybeans, rice, wheat. Those are all being studied to modify so that we can adapt them more quickly for the increasing global temperatures or for the increase in drought and extreme weathers. So we could take some of the traits that we know some plants have, like the ability to survive a drought. We could import that into a plant that is used for commercial crop like soybeans or corn, and increase the yields of those even during years that we have droughts. Livestock are also, of course, a tempting target for CRISPR because we could prevent them from becoming diseased. Which is one of the main costs of raising livestock, is that you are required to medicate your entire crop a lot of the time, and that's very expensive. Also, there's lots of excess antibiotics that are a pollutant to the environment around them. So there's a very huge benefit there that we could get some of these meat to those viruses by increasing their immunity, increasing their natural immunity. So as you can see, CRISPR has shown a lot of benefits and maybe some drawbacks. Hopefully you've enjoyed this video, and we'll discuss it more in the next one. Thanks. Have a great day.